Can you hear me? Yeah, perfect. So I'm Jonathan Sobel. I'm currently a postdoc at University of Lausanne. But today I'm not going to talk about my work. I'm going to talk about my hobby, namely sequencing beer DNA. So we had very great talk yesterday. Those were very technical. Here the philosophy is really to try to communicate with a very broad audience. So all the concepts are very simplified, and this is addressed to a very general public. So let me tell you a bit more about the context of this project. So basically, is it possible to do science outside of academia or industry? In an association or in a biohacker space, for instance? So the short answer is yes, but of course you have many constraints. You don't have the same infrastructure. It's difficult to get, for instance, enzyme to perform a qPCR or stuff like that. This project has been initiated in a vibrant community, namely Aquarium. So it's the biohacker space in Lausanne. It's a 3-year-old association, and our aim is to develop biology and more generally science-oriented projects, bioart, education, workshops. We are doing outreach, and of course we are implicating citizens, so non-scientists, for instance designers, and as well engineers and so on. So as I just said, our main concern is really to communicate among the association with people with different background, different knowledge, but more generally to communicate to the general public. And basically, why is it important? Our regulation, at least in Switzerland, is voted by the general public, and they need to know about really sequencing, for instance, or in order to vote the regulations or even the credits for this type of project. Why beers? Because I love beers. I must say, I drink a lot of beers. And more than that, beers is a fermented product. It contains different elements, such as yeast, hops, and even water that may contain bacteria. And all these different ingredients may contain DNA. And of course, there is a huge variety of beers, and this is why it's kind of interesting to study the DNA inside of the commercial and the craft beers. Basically, we aim at mapping beer at a molecular level to provide an open knowledge base to help drinkers to choose their favorite beers or to discover new beers based on our analysis. But we want as well to help homebrewers to define new recipes, to create new beers, and microbreweries, for instance, for their quality control analysis. So all this knowledge base can be used for that. With this project, as I said, we went outside, we went to festivals, we went to a conference, and we tried to communicate with the public with a cargo bike, where we were testing beer directly, and we were discussing, for instance, the PCR and why these DNA data are useful and important, and for instance, of course, how can we exploit the information of the beer DNA, but in a larger way, why their own DNA data are critical, and there are a lot of ethical issues around that. And it was a way to really start discussing with them and telling them about really the new technology and so on. Money, okay, this is great. You know that in academia, basically, you write a grant, you forget about it for 3 months, and then you get a negative answer. Basically, that's what happened for us as well. And of course, it was more difficult because we are not a recognized institution, so funding agency did not trust us and didn't want to give us some money, so we had to adopt another strategy. So we did a crowdfunding campaign. And this experience was very interesting because we had to communicate about the whole project, about the goal of the project, about how we will handle the data, what we will do with this data from the very beginning. And we had to do a media coverage in order to get bakers for this project. And 2 years ago, we succeeded to gather 10,000 Swiss francs to start the sequencing of beers. So it's time for Beeromics. Basically, our workflow was that the people who baked our project on Kickstarter were able to nominate a beer, and then we were sampling it with the community. We were analyzing DNA, so isolating DNA, performing PCR for ITS sequence, and then we performed the bioinformatics analysis. So here I show a bit our dream setup. So it's with the bento and with a MinION sequencer. Unfortunately, till now we don't have a MinION sequencer. So we were working with the facility, the genomic facility from the University of Lausanne for the actual sequencing part. But all the sample preparation and the library prep were done inside our hackerspace. Then we performed as well workshop to explain to people how to extract DNA from their favorite beers, and it was very fun to do that because of course we were drinking a lot of beers during this process. So it was really nice. And after that, I'm the bioinformatician of the project, so I performed a short read alignment with an ITS sequence database. So basically, just for the technical details, I built an index with BWA with this ITS sequence database, and then I simply aligned the FASTQ, the reads from the beers to this ITS database. So it's a very straightforward pipeline. It's the simplest I could do. And I guess it's the easiest one to understand, so there is no fancy algorithm behind. It's just pure ITS count per beer. So for the results, at the end we succeed to get 39 beers sequence with our targeted approach. You can find the raw data in this SRA database using this identifier, and all the methods and the quality control and the process tables and the protocols are in our GitHub repository. So if you want to have a look at this data, please do it, and come to me, and we can discuss. Maybe you can test some of your pipeline. For instance, there was this presentation of yesterday, yet another metagenomic tool. So that could be, for instance, a nice data set to play with. And then about the results, so basically in the A panel you can see our list of beers and the variety of ITS that were present in the beer. And we have a group where we have more than 40 ITS that were detected. And interestingly, this group of beers are self-fermented beers, like lambic, so with a less controlled fermentation process. And it's kind of funny to see that the variety of fungal species are more important in those beers. Then in the B panel, you can see all the detected ITS with so the different fungal species with more than two, so detected in more than 2 beers, and we have a whole group of Saccharomyces that are present in almost every beer, and we have some other species that are really specific to 1 or 2 beers. So that's very interesting, and that can help to trace really where the beer comes from, and to have an idea of maybe the impact on the taste. So that's one of the perspectives of the project. Then through this iterative process with data analysis, beer thinking, drinking, data analysis, and so on, So it was very intense for me. I came to this nice visualization where you can see all the beers in the y-axis and all the detected ITS, and the different colors represent the detection level, so the number of reads per ITS. And this allows to cluster the different beers together and see which one are somehow related based on their DNA content. And with that, I want to wrap up. And so basically, we succeed to sequence 39 beers that improve our knowledge about beer DNA content. The idea was to trigger a discussion about molecular biology techniques such as PCR and sequencing, and we could evoke, using this ambassador project, the ethical issue regarding DNA-related data. For the perspective of this project, we would like to involve other citizen science labs worldwide to sequence their own beers, or even people that are in academia and have some space in their sequencing machine, so maybe one lane, and they could load some of their favorite beers and then upload their data on a repository, and there we could grow our knowledge base about beer DNA. Then we want to investigate other markers like 16S RNA sequence. We want to do as well whole metagenome sequencing, potentially with a MinION if we succeed to get one. And of course, we want to develop this beer knowledge database. We want to help microbrewer to do their quality control. And later on, we want to explore the relationship between the beer DNA content and the taste. With that, I will finish my talk, and I want to acknowledge the BeerDeCoded team, especially Gianpaolo Rondo, who was the main leader of the group and who really initiated this project, as well as Luc Henry, who funded the Aquarium hackerspace 3 years ago, and the other people involved. As well, our baker from Kickstarter, the Vitality Cluster, and of course the OBF committee for awarding me with the OBF travel grant. Thank you very much for your attention.